Good morning, everyone. Good morning, Dr. Likewise. Welcome to my presentation. Today, I'll be talking about how I'll be cloning a bacterial expression vector containing my desired gene of interest and how I'll be transfecting a mutated cell line using this expression vector. My gene of interest is the BRCA1 gene, the breast cancer 1 gene, and the functions of this gene includes that it helps to repair DNAs and it is involved in pathways responsible for transcription regulation, chromatin structure regulation, the cell cycle regulation, and entirely genome stability. Mutations in this gene can cause breast cancer and ovarian cancer. My aim is to prepare my expression vector, making sure it contains a multiple cloning site, his tag for protein purification, the T7 promoter for strong expression, and restriction site, the NDI and the XHOI. And I'll also go ahead to transfect the mutated cell line that has a BRCA1 gene knockout. So after my vector selection, I'll go ahead and amplify up my gene of interest and also prepare my vector. I will select a PCR primer that is compatible with my vector, making sure it contains 5' NDE1 and 3' XHOI. I'll also use my PCR hot starter to amplify up my gene of interest and making sure I maintain PCR protocols diligently. I will digest my vector using the XHOI and NDI to linearize it, and also include restriction sites in my PCR product. I will send it off for jet electrophoresis to confirm for linearization and amplification. I will do my DNA ligation. I will ligate the BRCA1 gene and into the linearized vector using DNA ligase. After the ligation process, the next step is my bacterial transformation and validation. I already have my vector containing my gene of interest. I'll go ahead to transform top 10 E. coli cell using conditions necessary for transformation. I'll make sure I incubate on ice for 15 minutes, heat treatment at 42 degrees for 30 seconds. I'll also make sure I add my SOC medium, and then again I'll go ahead to prepare my agar plate that will be containing kanamycin, conferring the antibiotic resistance in my vector. This will enable me for the selection of E. coli cells that picked up my plasmids. I will incubate at 37 degrees overnight. I will go ahead to conduct cell lysis and protein purification. At this stage, I will be inducing the expression of my BRCA1 protein by addition of estradiol, and which will induce the T7 promoter in the vector. I will also go ahead and do Western blotting and to confirm for this process, and that's the validation process, and also send it off for Sanger sequencing. So here are the details involved in selection of the transformed bacterial cells. After the bacterial transformation, selection, and validation, we come to the final stage, and that's the transfection process. In this stage, we'll be transfecting our HCC1937 cell line that has a BRCA1 gene knockout. Our aim will be to insert our gene of interest into the cell line to correct the mutation. So we'll be using the pofectamine for our transfection, and afterwards we will run a functional assay to certify that the correction, the insertion of the gene of interest was properly done. And in this functional assay, we can see that our mutated cell line reveals no BRCA1 gene, while our clones reviewed our BRCA1 gene. We checked for their survival rates, and we also compared the repaired DNA, the repaired HCC1937 cell containing the BRCA1 gene. We compared it with the one that was not repaired. We also did a FACS analysis and did a comparison between the mutated cell line and the cell line containing our BRCA1 gene. Thank you. Here are my references. Thanks for listening.